Welcome to this class on Neuroscience of Human Movement. In this class, we will be talking about pathways within the basal ganglia. So, essentially, there are three pathways in the basal ganglia. So, in this class, we will be seeing direct pathway, the indirect pathway, and the hyperdirect pathway. Before we discuss the details of this, I think it is important to realize that these present some model of basal ganglia function. This is not a complete description of basal ganglia function. So, a lot of function of basal ganglia can be explained with the help of these pathways or the direct indirect pathway model. However, there are exceptions to this rule. So, one has to be careful while discussing or interpreting data from actual experimentation or from actual disease conditions, all right. However, in spite of the limitations, the direct and indirect pathway and the models associated with this continue to be relatively dominant. Let us remember that the striatum is the major input structure of the basal ganglia, and the substantia nigra pars reticulata and the globus pallidus internal are the major output structures. Here we have taken only the GPI as the output structure, and here striatum is called together separately as caudate and putamen. Important to realize is the fact that the thalamocortical neurons in the thalamus are excitatory and they excite the cortex. These thalamocortical neurons themselves are inhibited by the output from the GPI, by the outputs from the GPI. From here there is a very strong and tonic inhibition. That means there is going to be a constant inhibition of the thalamocortical neurons from the globus pallidus internal, or you can say that substantia nigra pars reticulata. For other neurons, substantia nigra pars reticulata also sends tonic inhibition to the thalamus or maybe to other output structures. So, basically essentially this defines an important function and an important aspect of basal ganglia function, that is the globus pallidus internal and the substantia nigra pars reticulata contain neurons that are tonically inhibiting the thalamocortical or the output neurons. So, because of this reason, so they are tonically means what? Tonically means constant inhibition. So, it is always present except when that neuron itself is inhibited. So, there are these neurons. So, these are GABAergic. These neurons in globus pallidus internal are GABAergic. Globus pallidus internal, from there these GABAergic neurons project to the thalamus and constantly inhibit. We could start explaining this by starting with the cortex or substantia nigra pars compacta, but I think it is better to start from this point, because once we catch this aspect, everything else becomes clear. So, important to note is that the GPI neurons are constantly inhibiting or tonically inhibiting the thalamocortical neurons. Now, it turns out from the striatum there are, so striatum means, so when we discuss the striatum we discuss, so let us say that is putamen, that is GPE and that is GPI. Let us remember that GPE forms part of the intrinsic circuitry, GPI is the output structure, whereas putamen is the input structure of the basal ganglia. The direct pathway involves projections from the striatum directly to the GPI. So, here you have direct projections from the caudate and putamen to the GPI. This means what? This is the input structure, this is the major input structure, and this is the major output structure, and the connection from the input to the output structure is GABAergic, mediated by the output of the medium spiny neurons, and uses GABA as the neurotransmitter. But let us remember that already GPI is tonically inhibiting the thalamocortical neurons. So, if these neurons in the GPI themselves are transiently inhibited by the input from the striatum, then what happens? For a brief period of time, the inhibition provided by the GPI neurons is halted. So, let us consider that that is the inhibition level, okay. That is the level of inhibition that is provided by the GPI neurons. Let us suppose at a particular point in time, at that point the striatum sends input directly to GPI and inhibits the GPI activity. Then what happens is that at that point the inhibition of the thalamocortical neurons comes down and then it continues again. So, during this brief period, the thalamocortical neurons or the excitation levels of the thalamocortical neurons increases, or the probability that they will get excited is higher because they are not getting tonically inhibited. So, essentially what you are having is an inhibition that is tonic, and that tonic inhibition itself is transiently inhibited. Essentially this is the activity, this is the most important function of the basal ganglia, that is disinhibition. Inhibition is provided by the globus pallidus internal segments, and that inhibition by itself is inhibited by the input, direct input coming from caudate and putamen, or from the striatum. Now, that essentially disinhibits transiently. Why? Because striatal inputs to the globus pallidus internal is transient, whereas globus pallidus input to the thalamocortical neurons is tonic, right. So, this inhibition is tonic, very important to remember. The inhibition from GPI is tonic. So, whenever striatum sends an input, for a brief amount of time the thalamocortical neurons are disinhibited, because the input, the connection to the globus pallidus internal comes directly from the striatum. Because of this reason, this is called as the direct pathway. Is there any other way in which it can happen? The answer is yes, there is another way in which it can happen. We will also discuss that. Okay. So, but what makes the caudate and putamen to disinhibit or to provide transient inhibitory input to the globus pallidus internal? That is the question. That is because cerebral cortex, actually through several pathways, not just the motor pathway, there are multiple pathways, association pathways, etc. Through multiple pathways, cerebral cortex sends glutamatergic inputs, that means excitatory inputs. Very important to note the signs here. Signs means SIGN sign, whether a connection is excitatory or inhibitory. Cerebral cortex sends excitatory or glutamatergic inputs to the striatum. Note, the striatal neurons themselves are inhibitory, they are GABAergic. So, when they are activated, they are going to produce GABA at their output. So, when they are excited, they are going to inhibit somebody, but they are inhibiting neurons in the globus pallidus internal, which by itself is already inhibiting the thalamocortical neurons. So, the cerebral cortex sends excitatory inputs, these are glutamatergic, right. So, or they use glutamate as the neurotransmitter in many cases, okay, with some exceptions. So, and the other strong input, the source of other very strong input received by the striatum is from the substantia nigra pars compacta, which sends a very strong excitatory input to the direct pathway through the neurotransmitter dopamine. Dopamine is the brain's pleasure chemical, one of the most important neurotransmitters in the function of basal ganglia, right. So, we will discuss its function in much greater detail in future classes. So, dopamine excites the neurons in the striatum. Once again, these striatal neurons themselves are inhibitory, and so that means they are going to inhibit the GPA neurons, which themselves are already inhibiting the thalamocortical neurons. So, essentially whenever the cortex wants excitation, it excites the striatum, thus causing disinhibition of the thalamocortical neurons. This is one important nice model or hypothesis of basal ganglia function. It is able to explain basal ganglia function and dysfunction. Okay. So, once again let us go through this. So, this is the reason we started by explaining from the tonic inhibition of the GPA neurons. So, GPA neurons providing tonic inhibition to the thalamocortical neurons is the most crucial element that you should remember. If this tonic inhibition is halted briefly, that means that there is going to be disinhibition, right. So, like here for example, like it has been shown here, right. So, and this inhibition is caused by inputs coming from the striatum, directly coming from the striatum to GPA. That means it does not go through the internal circuitry, the GPE, it directly goes to the output structure GPA, right. So, whereas that striatal neurons themselves are excited by two possible sources from the cortex. The glutamatergic projections from the cortex excite the striatum, and the dopaminergic projections from the substantia nigra pars compacta excite the striatum. This pathway from the cortex to the striatum, of course called as cortico-striatal pathway, and this pathway from the substantia nigra pars compacta to the striatum is of course called as nigrostriatal pathway. This pathway, so that is the nigrostriatal pathway, and that is the corticostriatal pathway. Okay. Why is this called as the direct pathway? Because the input from the striatum directly goes to the output structure, which is the GPI or the substantia nigra pars reticulata. Okay. This is the reason why it is called as a direct pathway. Then what about the indirect path? Another structure, the subthalamic nucleus, is involved in this. It turns out that the subthalamic nucleus provides excitatory inputs, glutamatergic inputs to the GPA, whereas it is tonically inhibited by the input coming in from the globus pallidus external segment, and the globus pallidus external segment itself is inhibited transiently by the input structure or the striatum. So, in this case striatum, which is the input structure, is connected to GPI, which is the output structure, through two possible intermediate connections. This is not direct, but rather through the globus pallidus external segment, which is part of the internal circuitry, and the subthalamic nucleus, which is part of the internal circuitry. So, these two things together make up the internal circuitry in this particular case. And so since the input is connected to the output not directly, this is called as the indirect pathway. The tonic input from the GPA to the thalamocortical neurons, that is transiently inhibited by the GPE, but the most important explanation actually comes through the pathway that involves the subthalamic nucleus, through that pathway. So, the GPI neurons are transiently excited, not inhibited, transiently excited by the subthalamic nucleus. So, whenever the subthalamic nucleus is activated, whenever it is excited, it is going to increase the inhibition level provided by the GPI to the thalamocortical neurons. So, that means whenever this whenever this block is active, the level of the inhibition provided is going to actually go up, right. But then this activity is inhibited tonically by the GPE. The activity of the subthalamic nucleus neurons are inhibited tonically, not transiently, tonically by the globus pallidus external segment neurons, which themselves are transiently inhibited by the caudate and putamen, by the striatal neurons. So, inputs coming in from the striatum briefly, transiently halt the activity or the tonic inhibition provided by the globus pallidus external. So, that means what? Essentially, inhibition provided to the subthalamic nucleus is reduced whenever there is a positive input that is coming in. Essentially, that causes an inhibition of the globus pallidus external, and that means the level of inhibition provided by the globus pallidus external segment to the subthalamic nucleus here reduces. That means the probability or the excitation level of the subthalamic nucleus increases, essentially then increasing the input to the globus pallidus internal, and that is not an inhibitory input, that is an excitatory input, essentially increasing the tonic inhibitory level provided by the GPI to the VAVL complex. When would this happen? So, this happens in situations when different from that discussed in the direct pathway. So, these are not the same cortico striatal neurons. These are a different set of neurons that are used conveniently when we do not want to do something, right. There is a need for a greater inhibition, right. In such occasions, there is an excitation provided by the cortex that increases the activity of the striatal neurons. The striatal neurons themselves are inhibiting the GPE neurons, and the GPE neurons themselves are tonically inhibiting the subthalamic nucleus neurons, right. So, essentially there is 2 minus. So, there is 1 minus here and there is 1 minus here, that becomes plus. There is a plus here, there is a minus here, there is a minus here. So, the output at that point is going to be plus, and that is multiplied by 1 more plus. So, that is an increased excitation level, and that is multiplied by a minus. Essentially, increased inhibition is the output of the indirect pathway, right. Why? So, the increased excitation level provided by the cerebral cortex increases the probability that striatum is going to inhibit the GPE neurons, and the GPE neurons themselves are already inhibiting the subthalamic nucleus neurons. So, there is 1 plus and 2 minus, essentially making the situation here the probability that the subthalamic nucleus is going to get excited to go higher, right. And the subthalamic nucleus is actually having glutamatergic outputs to the globus pallidus internal segment, and the globus pallidus internal segment is tonically inhibiting the thalamocortical neurons. Essentially there are 3 minuses, right, and 2 pluses. That means that net output is actually a minus. Something else is also there, that is what happens here. Here you have a different situation. Dopamine, which I said is usually an excitatory neurotransmitter, in this case acts as the inhibitory neurotransmitter for the indirect pathway. The details of this we will discuss in a future class, but please see the direct pathway. Here I said dopamine has a plus sign, but in the next slide I said in the indirect pathway dopamine has a minus sign. Why is this? We will discuss in a future class, but essentially dopamine or the substantia nigra pars compacta input is always excitatory. The net output of dopamine on the thalamocortical neurons is always excitatory. That is why it is having an inhibitory effect on the increased inhibition producing indirect pathway. So, if indirect pathway is inhibited, that means the net excitation level goes up. So, dopamine always has a excitatory influence on the thalamocortical neurons, something that we must discuss in future classes. Then there is the hyperdirect pathway. In this case, the subthalamic nucleus is directly excited by projections from the cortex, right. So, there is direct connections from the cortex bypassing the striatum to the subthalamic nucleus, and let us remember what the subthalamic nucleus does. The subthalamic nucleus is providing transient excitatory input to the globus pallidus internal segment, which provides the tonic inhibition to the thalamus, is it not? So, it is going to inhibit directly or super directly the output of the thalamocortical neurons by increasing the excitation level of the subthalamic nucleus. Note that in this case the major input structure of the basal ganglia, which is the striatum, is bypassed. So, here what you have is cortex. From the cortex there are projections to the striatum. In this case the putamen is taken as a striatum. This is putamen, this is GPE, and this is GPI, as in this is GPI, this is thalamus, this is subthalamic nucleus, right. So, the red connections here refer to excitation, and the blue connections here and the blue arrow marks here refer to inhibition, all right. So, there is excitatory inputs from the nigrostriatal pathway and the corticostriatal pathway that causes an inhibition from the striatum to the GPI, and GPI itself is tonically inhibiting. This is tonic, very important to remember this aspect. If this aspect is remembered, that GPI is tonically inhibiting the thalamocortical neurons, then it is very easy for you to explain and understand basal ganglia function, right. GPI is tonically inhibiting the thalamocortical neurons, and that tonic activity is transiently inhibited by the excitatory inputs coming in from the cortex and the nigrostriatal pathway. So, essentially this direct pathway excites the activity of the thalamocortical neurons or disinhibits the activity at the thalamocortical neurons. So, that means from the thalamus to the cortex there is an increased level of excitation. So, that happens, an increased excitation level happens. So, the possibility that there is going to be excitation at the cortex is increased by the activity of the direct pathway. So, in the indirect pathway what happens is the subthalamic nucleus is involved. Let us remember, so this is the subthalamic nucleus, this is the thalamus, this is the globus pallidus internal, and this is the globus pallidus external, and this is the putamen, right. So, in this case what we saw is that the subthalamic nucleus excites the globus pallidus internal, right. So, that is what we saw. So, essentially the input coming from the corticostriatal neurons continues to be excitatory. However, note the color of the arrow coming in from the substantia nigra pars compacta, that is actually blue, that means it is inhibitory. So, the corticostriatal neurons excite the striatum, or in this case the putamen, and which excites the neurons from the putamen to the globus pallidus external, and these are inhibitory interneurons, right. So, these are GABAergic. So, that is why the line here is blue, and they also further inhibit the neurons in the subthalamic nucleus. Right. Note this connection from the globus pallidus external to the subthalamic nucleus is actually tonic, just like the connection from the globus pallidus internal to the thalamus is tonic. So, the activity of the subthalamic nucleus is increased for a brief amount of time by the transient input received by the GPE. GPE neurons are inhibited, and these neurons are tonically inhibiting the subthalamic nucleus neurons. Important to note is that the subthalamic nucleus itself is having an excitatory influence on the globus pallidus internal. So, that means essentially there is one minus here and another minus here, and these two minuses cause an increased probability of excitation of the subthalamic nucleus, increasing the activity of the subthalamic nucleus neurons. So, these neurons send a higher level of excitation not to the thalamus, it sends a higher level of excitation to the globus pallidus internal segment, thus increasing the tonic inhibition level provided by the globus pallidus internal. So, essentially the indirect pathway is an inhibitory pathway. So, whenever the indirect pathway is excited, the level of inhibition provided by the GPI neurons to the thalamocortical neurons actually increases. Note what I have not explained here is the important role of the nigrostriatal pathway in this, which is inhibiting the indirect pathway. So, I already mentioned that the effect of dopamine on the thalamocortical neurons is always excitatory. This is something that we will discuss in future classes. Okay. Again, in the hyperdirect pathway, the input directly comes from the cortex to the subthalamic nucleus, and from the subthalamic nucleus to the GPA the excitation goes, but from GPA to the thalamus the inhibition level is increased. Suppose for whatever reason there is an important need to inhibit or cause an increased inhibition, then the cortex directly contacts the subthalamic nucleus bypassing the striatum. So, this pathway is called as the hyperdirect pathway. So, once again this is another pictorial representation of the situation. So, you have the direct pathway coming in from the striatum to the GPA, the indirect pathway coming in from the striatum through the subthalamic nucleus to the GPA and thalamus, and the hyperdirect pathway coming in directly from the cortex to the subthalamic nucleus. So, in summary, we have discussed three pathways: the direct pathway, which is responsible for initiation of movement, which is an excitatory pathway; the indirect pathway, which is important for suppression of movement, which is an inhibitory pathway; and the hyperdirect pathway, which bypasses the striatum, also an inhibitory pathway. So, with this we come to the end of this lecture. We will continue this discussion in future class. Thank you very much.